Hello, AP Biology students. This is our unit. We're going to focus on the cell cycle mitosis and also on meiosis and life cycles. So, some of this you have learned previously in advanced biology. There will be some additional things that you are learning now in this course as it is relevant to the advanced placement biology test. So what you must know the structure of the duplicated chromosome, the cell cycle and the stages of mitosis, the role of kinases and cyclin in the regulation of the cell cycle itself. So ultimately, the cell cycle is a life of a cell from its formation until it's going to divide. Functions of cell division are for reproduction, for growth, and for tissue repair. So we know that some organisms will carry out cell division or a cell cycle, and they'll use that for reproduction, like bacteria cells. We know that multicellular organisms grow through cell division and tissue repair. If we get a cut, you know that the cells will divide to help repair that tissue. So here you can see different examples, reproduction in A, growth and development in B, and tissue renewal in figure C there. So when we're looking at a cell cycle, we're looking at chromosomes or the DNA of those cells. And when we look at the DNA of all cells or all of the cell's genetic info, we consider that a genome. Prokaryotes themselves, they have a single circular chromosome, often referred to as a plasmid. A single circular chromosome that is stored in the nucleoid region. However, in eukaryotic cells, there are more than one linear chromosomes, and it actually depends on the species that you are discussing to actually know how many of chromosomes that we'll have. We know that for humans, we have 46 chromosomes. So mouse have 40 chromosomes. A fruit fly, Drosophila melanogaster, only has 8 chromosomes. And there you can see those chromosomes in the cells pictured below there. So each chromosome must be duplicated before the cell divides. So if you're looking at the figure there, you could say that 2 duplicated chromosomes are what we call sister chromatids. And those sister chromatids are going to be attached at a structure called the centromere. If you look, here you can see a duplicated chromosomes. Each strand here represents a chromatid, and right there where the constriction is, that is what you call the centromere. If you look structurally at the chromosomes, you could see you have that double helix of DNA coming down. On the chromosome duplication, including DNA replication and condensation, now you have the 2 duplicated chromosomes, or sister chromatids, and each has that double-stranded DNA. Remember that mitosis occurs in somatic cells, and then in meiosis, we have the formation of gametes. So somatic cells are body cells. They are diploid in nature, so that means 2 of each type of chromosome. So they have 2 copies of the DNA. So for us, that's 46. They divide by mitosis. So body cells will divide by mitosis. Unlike sex cells, which are gametes, the sperm and the egg, they are haploid, represented by just the letter N and not a 2N now. And haploid cells each have 1 copy of the chromosome. And gametes are going to be formed by the division called meiosis. Humans have 23. So our diploid number is 46. Our haploid number is 23. So if we look at the phases of the cell cycle, the cell cycle itself includes 2 phases. You have interphase and a phase called the M phase. Interphase is the longest period, as you could see in the figure here, is the longest period that the cell will spend the majority of its life. And there are three stages in interphase itself: the G1, the S, and the G2. The M phase is where we actually have cell division take place. And cell division includes two stages. There is the mitotic stage, in this case mitosis, which will take place. And mitosis itself is division of the nucleus. And then the next phase is cytokinesis. And cytokinesis is division of everything else in the cell. So looking at them a little bit closer, phases of the cell cycle, the mitotic phases alternates with interphase, G1S, G2, mitosis, and then cytokinesis. Interphase makes up about 90% of the cell cycle. In the G1 phase, the cell grows and carries out normal functions. In the S phase, this is where DNA replication takes place. So the DNA will replicate itself, or the chromosomes get duplicated. And in G2, the cell is going to prepare itself for division. So in that phase, you'll also have some other cell structures duplicate themselves, like mitochondria or chloroplast. In the M phase, which is mitosis here, in mitosis, the nucleus divides. In cytokinesis, the cytoplasm, so everything else is going to divide. Mitosis itself includes the stages of prophase, metaphase, anaphase, and telophase. And you can see in the diagram down below the illustrations of each of those phases. So G2, what happens is the centrosomes, this is animal cell, of course, centrosomes with centriole pairs. You have chromatin that is duplicated. In prophase, chromosomes consisting of two sister chromatids. You have a centromere connecting them. You start to get the aster formation, so the centrioles begin to shoot out mitotic spindle. So you get early mitotic spindle formation. Fragments of nuclear envelope in prometaphase, because in prophase, the nuclear envelope disappears or begins to disappear. Also, you have the nucleolus, which will disappear because you want to expose the DNA. You have non-kinetochore microtubules that aren't going to attach to chromosomes. So non-kinetochore microtubules are going to push out beyond the chromosomes. And then you have kinetochore microtubules, as illustrated here, that are going to attach to the chromosomes themselves to help move them. And where do they get moved to? They get moved to the center of the cell in metaphase. So they get moved to what we call the metaphase plate, or sometimes referred to as the equatorial plate of the cell. If you were taking a cell biology class, it would definitely be the metaphase plate. The centrosomes at one spindle pole, and then centrosome at another spindle pole up here, and you have spindle going in all directions. And the reason for that spindle is eventually it's going to start to elongate the cell. So you have the chromosomes, which are going to start, sister chromatids, are going to be pulled from their centromeres and migrate to the poles of the cell. The non-kinetochore mitotic spindle is going to help elongate the cell, and that will allow for telophase and cytokinesis to take place. If you study a college genetics class, in prophase is actually divided into like 5 additionally named stages. So mitosis. Mitosis is a continuous process with observable structural features. And you're going to look at the chromosomes because the chromosomes are what enables you to identify what stage the cell is in. So chromosomes become visible in prophase. Alignment at the equator is metaphase. Separation of sister chromatids takes place in anaphase, and you form 2 daughter cells and allow for cytokinesis to begin in telophase, and then cytokinesis itself is division of the rest of the cell. So prophase and prometaphase, if you look in your textbook, there are diagrams with descriptions. Understand what's going on in each stage. I did kind of just go through all that with you. Metaphase and anaphase, look how the chromosomes are arranged. Look at the mitotic spindle attached to the kinetochores. and how those that are not the non-kinetochore spindle or elongating, and there's cytokinesis and telophase represented. Cytokinesis is cytoplasm of cell divided. So division of the cytoplasm. In the animal cell, you form that cleavage furrow, which is evident in the picture there. In a plant cell, you get the formation of that cell plate. So what happens in the plant cell, you get a bunch of vesicles that are going to line up, and they're going to deposit the materials to lay down a new cell wall. So here you have the cleavage furrow. Here you can see those vesicles lining up to form a new cell wall. Look at the difference here. Here we have because this is an external view of the cell, this was taken with a scanning electron microscope. Because this is an internal view of the cell, we use the transmission electron microscope to gain that evidence there. So here you can see animal cell division with the fluorescence. And then here we could see plant cell division. So take a moment and really go back to these two slides and just look at those pictures. So animal cell and plant cell again. So which phases of the cell cycle can you identify? We will do this in lab as well. All right, bacteria cells divide by binary fission. Basically, the steps of binary fission, 4 to 5 steps. Typically, chromosome replication begins up here. So you'll have the circular chromosome, which will be replicated. The replication continues. And then what happens is, here's where the 4th and 5th step differences. As replication finishes, what will happen is now those replicated chromosomes will start to move towards the poles of the cell. And then eventually what will happen is 2 daughter cells result. So, and cytokinesis will begin. All right, so controlling of the cell cycle. This is another point that you need to know. Basically, you have this control system which is going to stop and go, allow the cell to either continue in its life cycle or stop it and say, nope, things aren't right here. And we call those checkpoints. It's a control point where stop-go signals are going to regulate that cell cycle. And if you look, you have the G1 checkpoint. At the end of G2, you're going to have a checkpoint. And in meiosis, during metaphase, you're going to have another checkpoint. So the M checkpoint. So the major checkpoints is the G1 checkpoint. That is the most important because it's going to look at these control factors such as cell size, growth factors, and the environment of the cell. G0 completes the whole cell cycle. So, oh, no, wait, not G0. Go means it will complete the whole cell cycle. If it says stop, the cell is going to enter a non-dividing state known as the G0 phase. Some cells, nerve cells, muscle cells, stay at G0. Liver cells are called back from G0 and can continue through. In the G2 checkpoint, controlled by DNA replication, completion, DNA mutations, and cell size. So the G2 checkpoint is basically ensuring that the DNA was replicated and completed fully. It's going to check for any DNA mutations. The cell itself is quite spectacular in that it could go back and look at the DNA that was replicated and fix any possible errors. Now we know that as we get older, or as an organism ages, let's talk about the human organism. As we get older, the accuracy of that DNA checking is not as efficient. So it decreases with time. And that's how we see as we get older, we start to get the onset of some diseases, cancer being one of them. So uh, as cells are going to undergo division, you're going to have a really good accuracy of going back and being able to make those repairs to the DNA, if the repair can be made. But as time goes on, you slowly start to decrease that ability. And you also have one during the M phase, and the M phase is, like I said before, is going to be during the metaphase. And ultimately what we are checking there, or what the cell is looking at, is the spindle fiber microtubule attachment to the chromosomes at the kinetochore, which is the anchor sites. So you have the spindle fiber microtubules, and where they're going to attach on the chromosomes is that area called the kinetochore. And it's going to make sure that those chromosomes are going to be lined up. So when anaphase comes into play, and you start to move those sister chromatids to the poles of the cell, you're going to get an equal division of the DNA. So here you can see cell receives a go-ahead signal. If a cell does not receive a go-ahead signal, it gets to the G0 stage. The M spindle checkpoint, mitotic spindle at metaphase, there you can see what's going on. If you look, this diagram is a very good illustration. You can see the kinetochore microtubules. And of course, all of these are being highlighted right here in the box. The kinetochore, basically what that kinetochore is, it's a complex of proteins that are associated with DNA at a region called the centromere that allow that mitotic spindle to attach there. Internal regulatory molecules, you have these things called MPF activity and cycling concentration. Ultimately, fluctuations of MPF activity and cycling concentration, you could see them during the cell cycle itself. So during the M phase, you could see that the MPF activity is going to increase, cycling concentration will decrease as we enter into, there's my little cursor, as we enter into interphase, and then during M phase they both decrease again, increase again, and then as we come out of M phase, it starts to decrease, and we are at low levels during interphase. So ultimately, this is kinases. And kinases, you have both cyclin-dependent kinases, called CDKs, and basically these are protein enzyme controls within the cell cycle itself, active when connected to cyclin. So you have kinases and cyclin, and when the cyclin-dependent kinases are connected with cyclin, it's going to activate them. Cyclins themselves are proteins which are going to attach to kinases to activate them, and you're going to have levels fluctuate in the cell cycle, ultimately, which you could see there in the diagram. So looking at those regulatory molecules, internally, MPF is that maturation promoting factor. That maturation promoting factor is going to be specific cyclin CDK complexes, which allows the cells to pass G2 and go into the M phase. So if you look here, you have cyclin accumulation, and here you have the cyclin-dependent kinase coming in, and ultimately what you form there is that maturation promoting factor here in G2. As it will continue through, the cyclin is going to degrade, and the degraded cyclin will be released, and then that cyclin-dependent kinase will come back through and cycle in. It's a molecular mechanism that is going to help regulate the cell cycle itself. You also have the external regulatory factors. External regulatory factors are anchorage dependence, density dependent inhibition, anchorage dependence. Basically, here's where you can see the difference between normal mammalian cells and cancer cells. So in anchorage dependence, this is when you have a cell that's going to either anchor to another cell, or it's going to anchor to the extracellular matrix from which that cell is in. And that gives it its dependency, which helps regulate the cycle itself of that cell. So we know that if we think of our skin cells laid out in a glass dish, when all present, will stop dividing, or because they are all attaching. And then if you take some of those cells away, what will happen is they will begin to undergo division again. But they only get so far to just heal that wound, and they won't start piling on top of one another. That's what we see here in cancer cells. So in the cancer cells, you get a nice one-layer sheet of cells, but you lose that control mechanism, and the cells will keep dividing, and you start to get a mass of cells there. So external regulatory factors, the growth factor proteins are released by other cells to stimulate cell division. Density-dependent inhibition itself, crowded cells normally stop dividing. Cell surface proteins binds to the adjoining cell to inhibit any growth. In anchorage dependency, the cells must be attached to another cell or that extracellular matrix in order to divide. So cancer cells themselves, cancer is that disorder in which the cells lose the ability to control growth by not responding to regulation. It's a multi-step process of about 5 to 7 genetic changes for a human for a cell to begin to transform. It loses that anchorage dependency and density dependency regulation. So cancer cells ultimately fail to respond to some of those signals and will keep forming a mass. And we know that mass of abnormal cells is what we begin to call a tumor. A benign tumor is a lump of cells that remain at original site. A malignant tumor or invasive tumor is going to impair the functions of 1+ organs, and that's called cancer. Metastasis itself is when the cells separate from tumor and travel to other parts of the body via the lymph and the blood vessels. And you can see that here in breast cancer formation. So you see a tumor grows from a single cancer cell. The cancer cells invade neighboring tissue. Cancer cells then spread through the lymph and blood vessels to other parts of the body. And basically what happens is the cancer cells may survive and establish a new tumor in another part of the body. Cancer prevention, basically physical activity, diet, and weight all come together for cancer prevention. So anyone can get cancer, but there are ways to minimize the risk of cancer. Don't smoke, legal or illegal, includes hookahs, chew, and secondhand smoke. Use sunscreen or sun protection when out in the sun. Exercise and keep your weight at an ideal level. Eat 5 to 7 servings of fruit and veggies a day. Use screening preventative measures. For example, breast, testicular, and mole checks. So females will get a breast exam and also a mammogram at a certain age as a preventative measure for breast cancer. Gentlemen will get testicular checks, feeling if there's any abnormal lumps or bumps, and mole checks for skin. And actually, mole checks are important because at any age, you should be getting your moles checked and looking for any signs of irregularity in shape, discoloration, multicolored moles, all possible for types of cancer. Practice abstinence or use condoms to prevent some types of cancer related through sexual activity, and also have vaccines such as the HPV vaccine, human papillomavirus. So that's it for today. That includes the lecture for mitosis and that part of the cell division or life cycle of the cell. We will continue on then with meiosis and life cycle reproduction in our next discussion. Thank you, everyone, and have a nice day.